Hello, I'm Dave Gordon, the Sacramento County Superintendent of Schools. On behalf of the California Dyslexia Initiative, or CDI, I'm delighted to welcome you to the 2023 Fall PLN. As the project lead, SCOE is working in partnership with the University of California, San Francisco, UCSF Dyslexia Center. Our goal is to build capacity and resources across California's education systems to address the needs of struggling readers and students at risk for and with dyslexia. The CDI is doing this through specialized professional learning modules, an annual webinar series featuring experts in the field, developing a repository of tools and resources, and what you're here for today, statewide professional learning networks. Last year, the California Dyslexia Initiative hosted 2 professional learning networks with a variety of fabulous presenters who are experts in their field of supporting students with reading difficulties and dyslexia. Today, we have the honor of learning from Dr. Jason Yeatman, Associate Professor at the Stanford Graduate School of Education. Dr. Yeatman's keynote will address how does the brain learn to read, why do some children struggle, and what is the impact of evidence-based intervention. Following the keynote, we have a variety of breakout sessions for you to choose from. Topics include empowering English learners, lessons learned from a personal perspective, screening and early intervention, and an introduction to structured word inquiry. We hope you enjoy the PLN and thank you so much for being here to learn with us in order to improve reading outcomes for all of our students.
Thank you, Superintendent Gordon. Good morning and welcome to the Fall Professional Learning Network. My name is Tammy Wilson and I'm the project lead for the California Dyslexia Initiative. I'm so excited to learn with you today from our amazing presenters. As Superintendent Gordon said, this morning we will hear from Dr. Jason Yeatman. Dr. Jason Yeatman is an associate professor in the Graduate School of Education and Division of Developmental and Behavioral Pediatrics at Stanford University. Dr. Yeatman completed his Ph.D. in psychology at Stanford, where he studied the neurobiology of literacy and developed new brain imaging methods for studying the relationship between brain plasticity and learning. After finishing his Ph.D., he took a faculty position at the University of Washington's Institute for Learning and Brain Sciences before returning to Stanford. As the director of the Brain Development and Education Lab, the overarching goal of his research is to understand the mechanisms that underlie the process of learning to read, how these mechanisms differ in children with dyslexia, and to design literacy intervention programs that are effective across the wide spectrum of learning differences. His lab employs a collection of structural and functional neuroimaging measurements to study how a child's experience with reading instruction shapes the development of brain circuits that are specialized for this unique cognitive function. I am really excited to welcome Dr. Jason Yeatman, and I am sure that we will enjoy this presentation as much as his daughter's kindergarten class enjoyed pieces of this presentation. Welcome, Jason.
Thanks so much, Tammy, and really a pleasure to be here. Get my slides going. Okay, Tammy, can you confirm that everyone can see my slides?
Yes, we see your slides. Thank you, Jason.
Okay, great. Well, pleasure to be here, and I'm tickled that my daughter's kindergarten class made it into the introduction. I did, in fact, give a couple of these slides to a class of kindergartners around here. You guys get the full longer version of the presentation. So I'm going to be talking about research my lab's done on the neurobiological foundations of literacy and trying to answer some of these important questions. But I want to start off first with an overarching goal of our approach. So my lab does research at the intersection of education and neuroscience. And if there's one goal that we want to communicate, that we want to inspire a virtuous cycle between these two disciplines. I'm a firm believer that interdisciplinary research catalyzes discoveries that aren't possible within the confines of one discipline on its own. So for example, many of the studies in my lab start off looking like this, a scenario that many of you are familiar with. We do interventions with small groups of children, usually groups between 1 and 4 children with a trained teacher. And as a neuroscientist, I see this as a powerful tool to ask basic questions about brain development, to understand how a child's learning environment actually sculpts the development of specialized brain circuits. Now, this image here is not just a pretty picture, it's actually data from the study I'm going to show you. By the end of this talk, you'll be able to explain what these different colors and shiny lines mean. So on the other hand, I then also aspire to use neuroscience as a tool to predict learning and personalize education. I want to emphasize this is an aspirational goal. We're not there yet. We're hoping over the next decade or so that we can make discoveries based on brain circuits that allow us to better tailor instruction to individuals. I will conclude, if I have time, with one step in this direction, the development of the rapid online assessment of reading. This is an open source and open access assessment platform that's emerged from my lab's research. So the learning objectives for today's keynote are, first, to understand how the brain learns to read. Second, to be able to answer what is different about the dyslexic brain. How do we measure brain development? I'm going to spend a little bit of time introducing the measurements we make. Finally, can we change the brain through education? And then I'm going to end, if I have some time, looking at new innovations in screening and assessment that have emerged from this work. Okay, so I'm going to start off with an experiment. If I were there in person, I'd be asking everyone to report out some information to me in a second, but I'll let you do this from the comfort of your own home. So please start off by staring at this fixation point here. Okay, did anyone in the audience, though I can't see you all, recognize one of the words on the screen? Most likely many of you did, unless Zoom glitched for a second. So I flashed a huge page of text for about 100 milliseconds. This is about how long an adult will fixate a word when they're reading. And the point I want to emphasize is that for many adults, word recognition is unconscious and automatic. When you see a word, you can't help but read it. And this is an incredible achievement that has come from many, many years of instruction and practice with this skill to build up circuits that are so automatic that you can't help but read. So let's think about literacy in the context of evolution first. And I want to begin by having everyone consider how unique literacy is in the context of evolution, and what an incredible achievement it is that many of you are able to recognize that word in just a brief glimpse. So literacy is relatively new for our species. There's evidence of basic communication as far as 500,000 years ago. And with the emergence of our species, there's evidence of spoken language, grammatical language, communicating through spoken language. However, the earliest evidence of literacy is a few thousand years old. So the first written language that's been found is Sumerian cuneiform tablets around 3100 BC. And the point I want to make is that literacy is a brief blip in terms of the human species. Literate societies are only a few hundred years old at the most, meaning that this is a new cognitive ability that's emerged from human cultures. It's not ingrained in our DNA. It's not as if a pile of books in front of a child will allow this skill to emerge, right? There's big differences between literacy and spoken language. So as I'm going through this talk, I want you to think about what are the implications of this? Why is this point important? And what might we hypothesize is different about learning to read versus learning to speak? And I'm going to make this point by thinking about the uniqueness of literacy in the context of child development. So this is a picture of me and my daughter, who's now in kindergarten. And what I want everyone to appreciate is that the two people in this picture see fundamentally different things and continue to for years. So when I look at this page of text, I can't help but hear the sounds of language. Just as all of you, when I flashed that page of text on the screen, your brain was automatically encoding not just the visual images of the words, but the sounds and meanings of them. So when I look at this page, my brain is doing that automatically. I can't help it. And this is, an incredible feat that I hear what I see. For my daughter, even years later, when she's able to talk and walk and communicate proficiently enough to, for example, demand that I read her another book, she still looks at this page of text and the symbols on the page are completely meaningless. It's just pretty pictures. Now, even a little more recently, this is right before she started kindergarten, pretending to read to her younger sister. And still, she loved looking through books, but the print on the page, even though she could, communicate with great proficiency through spoken language, written language was still meaningless to her. So this is unique when we think about child development. Most aspects of child development kind of take a continuous progression, whereas literacy is this discontinuity. Children start gaining literacy when they're taught how to read. So the point I want to emphasize is that literacy was invented by modern societies. The brain is not pre-wired to read, despite the fact that my daughter had looked at many books for many years. She didn't begin learning to read until she started being taught how to read. So the neural circuitry of literacy is constructed in response to education. For those of you that are teachers on this call, I want you to appreciate that the work you're doing in the classroom is actually wiring up the brain to have a new cognitive function that evolution never anticipated. So brain circuits must be rewired to connect vision and language in a new way. And finally, when we think about it from this perspective, I hope you can appreciate that it may be no surprise that many children struggle with this. It's not a function our brains evolved for. It's complicated. It takes years of work and practice to learn. So it may be no surprise that this is difficult for many children, and this is what we refer to as developmental dyslexia. So I want to emphasize this point by getting into some brain imaging data. So let's imagine what is happening in my brain versus, at this point, my 1-year-old daughter's brain as we're looking at this book together. So of course, we open the book and we're staring at the first picture in the book. And what's happening in our brains at this point is actually quite similar. So signals from my retina, from my eye, are sent to the very back of my brain, the primary visual cortex. This is the first stop of visual processing. This is the part of your brain that starts extracting the simplest features from a visual image to work towards recognizing this image. So primary visual cortex analyzes the location of lines, the general shape, the orientation of different segments and colors on the screen. And what occurs over the course of tens of milliseconds is that signals move up this bottom part of your brain, starting at the back, going up the bottom surface, through a series of circuits that are involved in visual recognition. Now, what's interesting here is that this process of seeing a bunny rabbit, processing this image in our brains, and recognizing what it is largely similar in my adult brain and my 1-year-old daughter's brain. However, things are fundamentally different as soon as my eyes move to the text on this page. So in my brain, within this broad circuit for visual recognition, is a specific region that scientists have named the visual word form area that develops as we gain literacy skills and is specifically involved in word recognition. You can also think of this region, the visual word form area, as the interface between our brain's visual system and our brain's language system. So of course, we have these language regions that have evolved over hundreds of thousands of years to support spoken language. The visual word form area is the interface that allows visual information to be transmitted and converted into language information. So it's kind of the hub connecting these regions. So of course, I'm not just leafing through the pages, I'm reading out loud. And as I start reading out loud, my daughter's spoken language circuits become activated, just like mine were by the written language. So in other words, for me, written language is automatically plugged into my spoken language circuitry. Well, for my daughter, these circuits operate independently. The process of learning to read means building the connectivity between these brain systems, allowing for this automatic transformation of information from vision to language. So this automatic process of rapidly converting visual information to language information also depends on the bundles of wires that connect the brain together. So I'm going to be showing some images like this. These renderings that look like wires are representations of what are called white matter connections. These are your brain's wiring system that allows information to be rapidly sent between different circuits. One important point that scientists have discovered over the last decade or so is that there are a couple specific bundles of wires. You don't need to remember the name, but I will introduce you to them. One is called the arcuate fasciculus. This is a connection that subserves many aspects of spoken language, including processing of speech sounds or phonology. And the other is the inferior longitudinal fasciculus. This is a major pathway that takes in visual information from the back of the brain and communicates it up to many other brain regions, basically allowing visual information to be shared with the rest of the brain. These are two specific connections that have been shown to be critical for literacy and develop differently in children as a function of their literacy skills. Okay, so 4 points I want you to remember throughout this talk is first, that learning to read drives the development of specialized brain regions for word recognition. Literacy means that written words are plugged into the brain systems that evolved for speech perception. The process of learning to read means building these new connections between these systems. So in the literate brain, written words are processed like spoken words, and the connections between visual and language circuits are essential for this. I'll go into this point in more detail. Finally, brains are different. Learning to read is easy for some children and difficult for others. There's neurobiological reasons for this, and I'm going to cover them briefly in the next section of this talk. So let's dive into this point and ask, what is different about the dyslexic brain? But before we get into this point, let's think about what the brain must learn to achieve literacy. So if we think about the component processes involved in reading, first, your brain must rapidly recognize a complex visual pattern. Your brain did that in tens of milliseconds when I flashed that page of text on the screen earlier in this talk. And then it must convert this visual representation, this processing of the visual information, into an auditory code. It's basically converting visual information into the sounds of spoken language. Then once the brain has done this, it can relate the sounds to the meaning of the words. And there is a wealth of neuroscience evidence supporting these different processes. We can actually see in real time as a word is flashed on the screen that we actually see brain activation in auditory processing regions and regions that process sounds a couple hundred milliseconds after a word is flashed in front of you. So learning to read requires coordinating visual, auditory, and language processing. And I want to bring back this picture of the brain and make the point that efficient wiring is essential. So these arrows that I've drawn as schematics here, the efficiency with which information can be rapidly sent between these circuits is critical for how children learn how to read. So when we get back to this question about what is different about the dyslexic brain, it may come as no surprise that efficient brain wiring is critical for reading and also is one of the differences in the dyslexic brain. So what I'm showing over here in this rendering on the right is a picture of the reading circuitry in the human brain. As the outside of the brain dissolves away, you're seeing renderings of these two bundles of wires. Whenever you see a rendering like this throughout the talk, I want you to think of an analogy to this picture of a NASA supercomputer over here, where these bundles of wires in the human brain allow information to flow from one circuit to another, for visual and language and auditory information to all be associated together. Much like these bundles of wires in the supercomputer here allow information processed on one node to be rapidly sent to another node. Okay. And so this image here is a rendering based on data, and I'm going to describe how we make those. But if we actually zoomed in with a very detailed microscope and looked at what this tissue looks like, this is what's called an electron micrograph. It's a high resolution image of the actual tissue. And you can see that at a very fine resolution, the white matter actually does look like bundles of wires. And each of these individual wires or axons connects one brain region to another. So the white matter refers to the brain tissue that contains these connections. I'll be using these terms throughout: connections or white matter referring to this type of tissue in the brain. And as I mentioned, children with dyslexia show differences in white matter connections. So there's a wealth of research showing how the properties of the white matter, the integrity of these connections, develop in subtly different ways in children with dyslexia versus typical readers. So we asked a question of whether we can change the underlying wiring of the brain's reading circuitry through a targeted intervention. So the goal here was for us to notice we can follow children over time. We can see that there's subtle differences in the structure of their brains. We might worry that this is an issue that can't be ameliorated by education. And we wanted to investigate whether by intervening using an evidence-based intervention program, whether we could actually change the developmental trajectory of the white matter. So we ran a study with 34 children with dyslexia. They were recruited for an intensive summer intervention program. We used the Lindemood Bell Seeing Stars program here. I bolded the point intensive because this is a key point in the study. This was done over summer vacation. students were coming in for 4 hours a day of one-on-one training, 5 days a week for 8 weeks. I'm not going to go through the details of the intervention because there are that would be a whole talk in itself of the components of carefully tailored intervention programs. But one of the important points here is it's not just practice and reading. This is a training program that focuses on the building blocks of literacy. So skills such as segmenting and blending speech sounds, building new words and deconstructing words with different letter combinations, working on systematically building up to decoding new, more complicated words, and then a series of activities that are focused on building reading fluency. We had a control group of children going through business as usual so that we could compare the effects of the intervention to typical development over this time scale. We then brought these children into the lab every 2 weeks, and we made measures of reading with a series of assessments that I'll talk about that are familiar to many of you, to measure reading accuracy, reading rate, fluency, and comprehension. And then we made measures of white matter connections with a technique called diffusion MRI. So focusing, oh, by the way, I'll talk more about diffusion MRI before I go into the findings from this. But first, I want to focus on the findings in terms of behavioral data. So this is showing data on one participant in the study. This is a measure called the Woodcock-Johnson Basic Reading Skills Composite Index. This is a standardized measure of single word reading, so decoding skills. A score of 100 refers to the population average. That would be a child that's performing at the national average in terms of single word reading. And then this bar here is showing the normal range. So this is data on one participant who came into the floor of this measure. This was a 9-year-old subject in this study who came in at the floor of this measure, meaning that they were only able to read 3- and 4-letter words. So for those of you that work with students of this age, you know what a severe challenge learning is going to be across many different subjects for this kid who is struggling to this extent with reading. Over the course of the intervention, you see substantial improvement for this child, not quite getting up into the average range, but getting quite close. I'm going to show data on some different individuals. So here are 5 individuals that we've randomly selected. So you can see they start off at different places. All of them are struggling readers. Some of them are having severe, substantial struggles. Other are slow, effortful readers. But all of them are showing growth over time in this intervention. Here's showing the average growth for the whole group. And to summarize this, this is children going from, on average, the 10th percentile to the 30th percentile on measures of reading skills. This is about 1 grade level of change. Some of the children in the study increased as much as 2 to 3 grade levels over the course of summer. So tailored interventions can be highly effective for some students. We looked at a number of different reading measures, and we see significant growth over all the different measures of reading skills. So I'm going to show reading accuracy, shown in orange. That was what I showed you before, basically complexity of words that a child can decode. Reading fluency in blue. This is a timed measure of how quickly children can read for comprehension. And then finally, reading speed in green, which is just a measure of how quickly children can read. So we see significant growth across all these different reading measures. We see larger improvements in accuracy than fluency. And this is a typical finding that it takes more work to intervene and improve reading fluency, though it is obviously very possible based on these data. We still see large growths in terms of reading fluency and speed. So we might ask now, what about the children with the greatest struggles? What about the children who are showing the most substantial difficulties? So I'm going to show you a plot here where along this axis is each child's reading score at the beginning of the study, so before they started the intervention. Over on this side of the plot are the children with the most severe struggles, and over on this side are the ones that are slow, effortful readers, but not showing the same level of difficulty. This axis here is showing the amount of growth in reading fluency that each participant showed. So what you can see is that the participants who are coming in with the greatest struggles, the ones with the lowest reading scores when they entered the study, these are participants that are between the 1st and the 5th percentile on standardized reading measures, are actually the ones that are showing the most improvement. And to me, this is a big success case for intensive interventions, that even the students who are struggling the most are helped to a huge extent by evidence-based intervention programs, and even show the larger gains in terms of quantitative measures of how much reading improvement they show compared to children with higher reading skills. Okay, so now I'm going to transition into the brain imaging data to look at how the brain's actually changing in these individuals as they learn. Before I do that, I want to spend a brief moment asking the question of how we measure brain connections. I want to give you all a little sense of what it's like to participate in these studies and what these measurement techniques actually do. So everything we do in the lab depends on magnetic resonance imaging. Many of you have probably been in the same type of MRI scanner that we use for our laboratory research. We use the same MRI machine or scanner to measure many different properties of the brain and also other types of tissue. So I had my first MRI when I had a high school sports injury and had an MRI for a torn disc in my back. It's the same exact machine that we use to make these quantitative measures of brain development as well. So there's a number of different kinds of MRI. Structural MRI refers to a measurement of the anatomical structure of any part of the body. So in our case, it's always going to be the brain, but it could also be a structural MRI of the knee, or in my case, earlier in life, my back. Functional MRI refers to a measurement that indexes changes in blood flow that result from neural activity. So when I was showing you those images earlier of the brain on fire, of different parts of the brain lighting up, that is data that we've processed based on functional MRI measurements that are indirect measures of how blood flow changes in the brain as different parts of the brain become active, as different populations of neurons start working harder to process information. So functional MRI is this technique where you see bright spots in the brain that indicate which parts of the brain are processing information or becoming active on a specific task. Then finally, diffusion MRI, and this is the measure I'm going to be focusing on for the next part of the talk, is a measure of water diffusion in the brain. You might be thinking, why are we interested in water diffusion when we're interested in brain systems? Well, water is constantly moving around and probing its local environment. And it turns out that measuring water diffusion is a very efficient way to measure the connections between different brain regions. So let me give you an intuition of why that is. Why is water diffusion a good index of brain connections? So this technique, diffusion MRI, as I said, it's a completely non-invasive measurements. That's another important point to emphasize, that all of these MRI measurements are completely non-invasive. I've been in the MRI machine probably 100 times. I'm always the first participant in every research study that my lab has ever run. There are no health or counterindications of having MRIs. And so diffusion MRI is like all MRI techniques we use, it's a non-invasive measure that indexes the flow of water through brain tissue. Now, I want you to think of an analogy to pipes. So if we imagine that these white matter connections, remember I showed you that picture of these little tubes. These are the wires that connect different parts of the brain together. And yes, in fact, at the microscopic scale, they are little tubes. So we measure water diffusion to index how water is flowing through these tubes. And an analogy that's actually quite a good one you can think about is thinking about the insulation around these tubes or pipes of the brain's piping system. So in the case where there's a lot of water diffusion, you can think of that like a leaky pipe, right? A pipe like this where water is spilling out everywhere is one with less insulation, like less integrity to it. And as we measure a decrease in water diffusion, that means that that these tubes through the brain, these wires or the brain's piping, whichever analogy works best for you, are becoming better insulated. I am here in Silicon Valley. So for those of you where a internet analogy works better than a plumbing analogy, can also, you can think of these brain connections as, bundles of wires that send information back and forth. So another analogy you can think about is as we see a decrease in water diffusion, it indicates better insulation, more densely packed wires. You can think of that as going from simpler, let's say old copper wires to new fiber optic cables that are higher bandwidth. So the point I want you to remember throughout is that a decrease in water diffusion is like indexing an increase in insulation or integrity or a higher bandwidth connection. What does it actually look like to make these measurements? So of course, you're lying down in the MRI machine, and these are measurements of brain structure not brain function. So what that means is that we can make the same measurement no matter what you're doing. All you need to do is hold still in the machine. And to keep all of our participants happy, we always show them movies. Kids get to choose their own movies. When I first started this research, here is our most popular movie, SpongeBob. But we quickly realized that kids and adults as well can't watch SpongeBob without laughing. So more and more we've switched to Madagascar. And one of the difficulties about MRI is that you do have to lay still. So the nice thing is you can lie in the scanner, you can watch a movie, you can be quite comfortable as we make these measurements of the structure of your brain. Have to hold still throughout the course of the MRI scan, but we keep kids comfortable and they're able to. This is what the raw data looks like. You have fuzzy pictures of the brain. And one of the other components of my lab's research is engineering software packages to extract quantitative metrics of brain connections from this raw data. So we build a lot of tools. We make them open source and freely available to the public. They're used widely throughout the world for basically taking this raw data from the MRI machine, being able to trace these long-range connections, these bundles of wires across the brain. And then we extract measures of water diffusion for specific connections in each individual's brain. And that's what I'm going to be showing you next, is some measures of these specific brain connections in individuals as they learn. So the big point that we find is that interventions induce systematic changes in brain connections. So this is showing a zoom in of one individual's arcuate fasciculus. Remember, this is the pathway that I introduced earlier that connects regions of the brain that are involved in processing language information, particularly phonology or the sounds of spoken language. And here is the rate of water diffusion in this pathway at the beginning of the study. Now, this measure on its own is not that interesting unless you're a physicist who's interested in water diffusion. But what is interesting is how it changes over time. So this is showing this group of individuals when they first came in, and then 2 weeks later. So after 2 weeks of this intensive intervention program, we already see a highly significant decrease in water diffusion. And remember that a decrease in water diffusion is indexing better insulation, more densely packed wires in the brain. So in this group of subjects going through the intervention, we see a systematic decrease in water diffusion that happens very quickly within 2 weeks of starting the learning process and persists over the whole course of the intervention. So 0 hours being the beginning of the intervention, after 50 hours, so about 2 weeks, about 6 weeks, and then at the end of the intervention. And we can compare this to our control group who is not going through the intervention. What we see is that there's a highly significant decrease in water diffusion, the participants who entered this intensive intervention program, and no change over this short period of time without the intervention program. So bringing back this analogy, remember less water diffusion, or this decrease in water diffusion we're seeing here, indicates better insulation or a higher bandwidth connection. Basically, the tissue in the brain is more densely packed and more highly organized. So one big takeaway based on these data is that these intensive intervention programs are actually changing the structure of the brain circuit. We're not just teaching children a strategy to work around some intrinsic difference in their brain structure. The structure of their brains, the way these wires are bundled together, are malleable and over this time scale of weeks. So I focused first on this one brain connection, the arcuate fasciculus. I want to zoom out and get back to this initial slide that I showed you. That was actually data from this study. And what I want to emphasize is that we don't just see changes isolated to one small piece of the brain. We actually see growth throughout an extensive network of brain connections. So shown in this image here is a rendering of a bunch of different brain connections. I was focusing first on this one here, the arcuate fasciculus, but I'm showing a bunch of other connections between the visual cortex and between language areas, between different parts of the language network. And the color map here is indicating the amount of change that we see in each connection. So green ones are ones that are stable, that are showing no change, that are not showing plasticity or reorganization over the course of this intervention. As you move from red to yellow, these are connections that are showing greater growth over the course of the intervention. And the big takeaway here, and this was a surprising finding in the neuroscience literature and also an exciting innovation in how we understand brain plasticity, is that these brain changes are widespread. They're not just localized to a single isolated brain region. Furthermore, as we look at these changes in individual, we see that the growth trajectory, the week-to-week change in water diffusion for one of these brain connections is actually tracking that individual's learning process. So if you remember early on when I showed you differences in growth trajectories with some individuals rapidly showing improvement in reading skills and flattening out, others showing kind of more systematic week-to-week change, that we actually see every time we bring them in and measure reading skills that tracks the growth in these white matter connections. Okay. So a couple takeaways I want you to come away with from this part of the talk. First of all, reading difficulties are associated with differences in brain connectivity. I mentioned that early on. I didn't go deep into the data, but there's a wealth of data showing that when we do comparisons between individuals with dyslexia to individuals with typical reading skills, or we track development and look at the changes in connections over time. Now, individuals with dyslexia show subtle differences in the organization and the development of these white matter connections, these bundles of wires that connect brain circuits together. Many people have worried that that's an intrinsic difference that won't be amenable to intervention. We've shown through this research, and others have now replicated this finding. It's exciting that just in the last year, a couple of other labs have replicated the same finding, that even in children with severe dyslexia, interventions are highly successful. This is a really important point that based on our data and others, that even children who are really showing the most profound struggles can learn when given an appropriate evidence-based intervention program. Now, finally, getting to the brain imaging data, intensive interventions change the connection to the brain reading circuitry. And I wanted to bold intensive here because I think this is a key point, that children who really have profound struggles learning to read need intensive and evidence-based programs. And so we did this over the course of the summer, and because this was detailed work done in a lab, we were able to work for 4 hours a day, so have a large focus on reading skills. We don't know the exact dosage of intervention that's required here, but it is important to note that the dosage of the interventions, the amount and intensity, so the amount of hours per day and how many days a week, the amount of time that's focused on these skills is directly related to the amount of growth children show. So one of the, I think, most important takeaways, and I wish I was in a room in person, I suspect that many people that are listening right now have spent a career teaching. And I think as a society, we need to recognize teachers as brain engineers, that the work you all are doing in the classroom is sculpting the development of these young brains. When we think about teaching a child new skills or teaching them new information, teaching, unlocking, for example, literacy and mathematics, that this learning process is fundamentally sculpting the way their brain is wired together. This is an incredible thing. I mean, the work you're doing in a classroom, to me, is analogous to wiring up a new computer to do an incredible thing. So you can say this in another way, more broadly, that education is sculpting brain development. The experience that children are having in classrooms is driving the development of brain circuits in different ways, allowing these new skills to be unlocked. And what I hope is that over the coming years, we can have more research practice partnerships, connecting researchers with educators to do more detailed research on this process, on the exact ways in which education is sculpting brain development and how we can use neuroscience knowledge to contribute to the classroom. And to that last point, I think another takeaway is that we need better methods for early identification and intervention. For children, the earlier we can identify struggles means the earlier we can intervene. It means the less time that they have to spend struggling with reading skills. And it also means that we can intervene during a period of time when the brain is more plastic, more amenable to intervention. That's one of the big takeaways. The intervention literature is that you get more bang for your buck in terms of more growth in reading skills for the amount of intervention time when interventions are done earlier. So I spent most of my, yeah.
Okay, so there are a few questions in the chat, and you asked me to interject when I thought it was an appropriate time. So with those takeaways, one of the questions was, Is this kind of improvement true for kids of any age, or does it become more stagnant once they reach a certain age? So it kind of speaks to your early intervention question.
That's a great question. So I'm going to give a couple answers to that question. So first of all, in this study, we did look at differences as a function of age. And in this intervention program, we found that it was equally effective for the younger participants and the older participants. The children were between the ages of 7 and 12 in this study. And that the amount of brain plasticity that we saw was roughly equivalent in the younger and older participants. So that's a really important point, is that it's not for older children with dyslexia, it's not lost. Interventions are still highly effective. The brain is still plastic. Okay. Though I also want to emphasize that we worked in a unique context, right? We were able to work one-on-one, 4 hours a day, 5 days a week for 8 weeks. Other studies that have done intensive intervention programs, but without this opportunity to personalize the intervention to the extent that this was able to, find that there is a relationship between the age of intervention and the amount of growth that's seen. So across a number of studies showing that earlier interventions are more effective. So I want to summarize both those points, because it's a really nuanced and important point that interventions are still effective for older students. Older brains are still plastic. So don't give up on the older students that are still struggling with reading. But when you think about what's the best bang for your buck, early interventions are more effective. And I think there's another really important point. Sorry, Tam, I'm giving a very long response, but this is like one of the most important questions to pause on. So some people look at this data and say, okay, well, interventions are effective throughout. So, why worry about early identification, early intervention? As many of you know who work with children with dyslexia, having these struggles in school can cascade to every other aspect of school. From, as math goes from symbols to word problems, children with dyslexia can start struggling in math, even though they might have exceptional math skills. As soon as history classes are depending on reading books and extracting information, as soon as reasoning skills are taught by reading and analyzing text, suddenly a struggle with reading becomes a struggle across many other topics. So by choosing to not intervene early, we're choosing to let children have these struggles persist and go beyond just reading to all these other areas of their education. There also is, unfortunately, very sad data showing the mental health implications. And I want to emphasize, anytime we're talking about research findings, this is not every kid. So for those of you with children with dyslexia or that work closely with them and advocate for them, this isn't deterministic. But on average, children with learning disabilities tend to have higher issues of many other mental health issues. And find me offline sometime and we can have a longer conversation about this. I think it's obvious on how traumatic it can be for many students who go through school unable to accomplish this skill or struggling deeply to accomplish this skill that's so easy for their peers. So I think there's a number of reasons. Even though interventions are effective for older students, I'll say that again, don't give up on the older students with dyslexia, but there are many, many reasons why it's preferable to intervene early.
Great. Another question is around the most promising evidence-based intervention programs. And perhaps rather than call out particular programs, you could tell us what components we should be looking for in evidence-based interventions to support students with dyslexia.
Yeah, so I'm going to apologize and not answer this question, but I'm going to tell you why. That is a deep and long conversation in itself. We probably want to schedule a couple hours to talk through, the components of a successful intervention. And I don't want to do it the injustice of here's the 2 bullet points. What I will say is there are a couple kind of high level things that are important. And one is this structure. So when we're thinking about a child who has profound difficulties with reading, kind of interspersing, a little bit of phonological awareness here and a little bit of extra practice with fluency there is just not enough to help them overcome their struggles. Now, that's not to say for teachers here, don't do that in the classroom. I do what you can, but as an educational system, we have to think about structured and systematic, well-organized intervention programs that progress, that have a evidence-based progression through a series of skills that are woven together. Now, I realize that this is not giving you like the detailed information of, like, I'm looking at, for whoever here is looking at 5 intervention programs and trying to decide which one to work with. If you are in that case, I would say, this is, and I'll get to this in the last point of my talk too, in important connections to build between scientists and educators. Is that my hope would be that if you are in that decision-making point of which of these educational intervention programs to select, or you're a district leader and wondering about which approach to go to, that we need to make ourselves accessible to talk those issues through in more detail, beyond just giving you the here's my favorite intervention program, which I won't do, because there's a lot of good ones out there. There's also a lot of mediocre ones out there, and I don't want to do the injustice of like naming a couple good ones and not giving the kind of full list.
All right, thank you, Jason. I'm going to let you get back to your talk, and I just do want to say what you said about the structure matters and how it's implemented matters. So I think that's
yes, those are 2 key takeaway points. The idea of I'm trying not to use any of the buzzwords of the era, because but having programs that are systematic, structured, have a theory and evidence for how the different components are woven together is really key. Okay, so I focused on this part. I showed you a lot about how education, how intervention programs can help us understand basic principles of brain development. A lot of this was basic science, right? A lot of what I was telling you is basic principles of brain plasticity, how these connections are changing in real time as children learn. I hope you were able to take away a lot of the high level inferences that I think do matter for education. But now I want to spend the last 10 minutes here closing this loop. So as I said, my high level goal is to think about neuroscience as a tool to predict learning and personalize education. And I'm going to tell you about one step in that direction, the rapid online assessment of reading. This is an open source, open access, totally free online assessment program that we've developed and it's emerged from our research. It's not neuroscience in the sense that kids are not put into a scanner for this assessment, but it did come from the decade or so of research that we had done on the mechanisms of learning. So I'm going to switch over to a second short 10-minute talk where I'm going to introduce the Rapid Online Assessment of Reading, or ROAR. Our goal here, and I have emphasized this point throughout, but this is really the current focus in my lab, to build a bridge between the lab, the community, the classroom, and the clinic. And I want to emphasize that I'm saying a bridge between the lab and the classroom, and not a one-way street. And I want to start off by saying, when I think about scientists or academics developing tools that make their way into practice, be them technology or different approaches, that's really critical that this is done through a partnership model, where it's not me as a scientist inventing something and then sending it out into the world, but instead taking the knowledge of science that my lab and many others have developed, and working with educators, be those district leaders, teachers, reading specialists, to also make sure that the wealth of knowledge by those that are actually working with kids is brought into the development of these tools and the science that's underlying them. Okay, so the problem that we're trying to address here is that assessments are time-consuming, costly, and grounded in opaque proprietary products rather than open science. This has been a challenge in my own lab's research that really was highlighted right around March of 2020 for obvious reasons. So our solution, the Rapid Online Assessment of Reading, is, as I said, an open source, meaning that other academics can work with the underlying code, make improvements to it, change it, see exactly what the assessments are doing. Open access, meaning that any educator can use this tool, or school district can use this tool completely openly. And it's grounded in ongoing research, and it's co-developed with a diverse group of school district partners. So as I went through in the first part of the talk, this is kind of a schematic of the research my lab does. We study the interplay between brain development, plasticity, and learning. we develop interventions, we combine those with brain imaging measures. We're always trying to develop more detailed measures of behavior to understand, how reading is changing over time. Now, of course, many of you, hopefully all of you, probably, or maybe, I don't know, hopefully some people have forgotten, the world changed in a fundamental way in March of 2020. As we were locked out of the lab, we were at home trying to innovate and figure out new ways where we could continue our research without, during this era, being in the same room as our research participants. So we started developing an online platform called the ROAR, the Rapid Online Assessment of Reading. The idea was that this was going to be a completely automated assessment delivered through the web browser. And through a series of studies that I'm going to briefly talk through, we're able to achieve actually better reliability than most of the individually administered one-on-one assessments that we are using as the gold standard to compare ROAR to. The exciting research side of this is being able to pursue much more inclusive research. So this is an ongoing research study. It's still going in my lab. We're still expanding our partnerships. And for us, this is a big opportunity to reach broader and more diverse populations that are often excluded from research. I say jokingly often, but obviously not a joke, that the participants around my area of Stanford University really aren't representative of the diversity of young learners in this state. And by moving to online research, we're able to engage with populations of young learners that truly do represent the diversity of the students that we want to understand reading development for and understand assessment and intervention for. Our goal is to lift the burden of screening, assessment, and progress monitoring in schools by developing more efficient kind of automated tools for this that are both fun and engaging, designed for kids, and also provide useful and actionable data for teachers. So our method to do this is through research practice partnerships. So as I mentioned earlier, the way we like to work in these types of studies is try to think about a virtuous cycle between research and practice. So this means that we're embedding ourselves in schools. We're allowing our partners to influence the design of the research questions. It's not just us coming with a top-down idea of this is what should be done, but we really want the schools we partner with also to guide us on what are the pressing questions about reading development. Our mission is to keep this open source and open access and to pursue scientific transparency. So every aspect of the ROAR is published in peer-reviewed open access journals, and we try to summarize each of our publications on our website. You can visit our validation page to see the sequence of ongoing validation studies of this tool. And open access transparency is really at the heart of our goal. Right now, we have expanded to have research collaborations with over 60 schools around the country. This is in our local community, representing urban, suburban, and rural districts across 14 states now. We also work with many independent schools that focus specifically on learning differences and learning disabilities. And we're in a phase where we're now growing to try to support schools around the country. So we're starting to expand to working over the next couple months with 30 states and eventually internationally, though we're holding off on that for the time being. Okay. So let me tell you about the development of this tool. Our goal is to keep this grounded in our ongoing cognitive neuroscience research and make sure we validate it both in studies in the lab and studies in the school. So in our first prototype study, and this was done in the spring of 2020. We had just shut down our research lab. We had a large group of participants who had come through our research lab and been administered one-on-one assessments. So we had a group of subjects where we had Woodcock-Johnson basic reading scores. When our lab was home working over Zoom, we spent the first couple months developing a prototype of a task that we think would index word recognition quite accurately without requiring a test administrator. So we ran this first study, and what we found, what this plot is showing, is the percent correct or proportion of correct responses on our first prototype version of the ROAR, and how that correlates with individuals recently administered Woodcock-Johnson reading scores. So what we found is a very strong correlation of 0.9, meaning that this quick online task was a very accurate index of this individually administered assessment. Our next worry, though, was to what extent a automated unproctored technology-based assessment would be accurate in young children who often struggle to follow instructions and interact with technology. So this next study was run where we administered the Woodcock-Johnson over Zoom. We then made a shorter version of this of the Roar assessment. We made the characters more engaging. We had the instructions, the tasks are narrated by different fun characters. We ran a validation study in a group of 6- and 7-year-olds. So the color of each of these points is showing the age of the participant. On this axis is showing their score on the rapid online assessment of reading versus their Woodcock-Johnson score. And once again, we had a very nice correspondence between these two measures. This was quite exciting to us that we were able to make the task engaging enough to get accurate data in even the youngest learners here. So then our next validation study was done when schools went back into session. This was like early after the return to in-person learning, kind of showing you the trajectory of development and validation of this tool. So we partnered with some school districts in California. We deployed the Roar in 18 first-grade classrooms. And we compared that to the assessment that was being used, standard of practice, which was an individually administered assessment. And what we found is that we were able to very accurately classify who is flagged as struggling based on the individually administered assessment with this automated tool. So the next series of studies we did were to optimize the reliability and make sure it's efficient, fun, and engaging for young kids. So the first thing we did was optimize it for reliability, which means that if a student takes the assessment twice back to back, we want to make sure they're getting the same score each time. So this is showing the relationship for a couple hundred students between their performance on one form of the assessment versus another. We're able to achieve quite good reliability. More recent work that actually just came out a couple days ago. Actually, we just posted the preprint on a preprint server, meaning that you can all go and download it if you're interested in the details. We implemented a computer adaptive version of this assessment. And we ran an experiment where participants were randomly assigned to a condition where they got it using the computer adaptive version versus the more standard version. What we found is that we were able to achieve this very high reliability greater than 0.9, in a much faster assessment by using this computer adaptive testing algorithm. Now, right now, as part of our ongoing research is around the user experience and trying to design characters and gameplay that's engaging and fun and validating for young children, but also maintains the psychometric properties, so maintains the basic technical aspects of the assessment. So what I was describing was our Roar single word recognition measures, so a measure of single word reading abilities. We now have a suite of measures, including phonological awareness, sentence reading efficiency, and vocabulary. You can demo all of these openly on our webpage, and the tool is open for broad use through a research practice partnership model. So I'm going to conclude there. I want to, a couple concluding points is that through careful design, validation, automated online assessments can achieve similar reliability and validity as standard individually administered assessments. And as you can tell, hopefully throughout the course of this talk, the first half on the neuroscience part, this final part on new measures we've developed, is that research practice partnerships are at the foundation of how we work. Our goal through this model, how we have felt is that our partnerships with school districts are inspiring new research directions. They're inspiring us to tackle research questions that we would not have thought of sitting in the confines of our own lab. And hopefully, and this is aspirational, is that this leads to tools that are grounded in real world problems. So grounded in the challenges that many of you face in the classroom, as opposed to what we might think are the challenges from the view in our laboratory. And hopefully I'll conclude at this point, we can realize the broader goal of building deeper systemic relationships between research and practice to support the diversity of learners in California and beyond. So I'm going to end there. I want to thank you all for, well, I guess not joining me in person, for joining me over Zoom, and also the many incredible folks, partners and funders. These are some of the districts we've worked with, some of the organizations that have generously funded this work, and then most importantly, the many individuals who have actually been out on the front lines doing all this work. So thank you.
Thank you so much, Dr. Yeatman. You have given us a lot to think about, and I am so excited to be able to learn from you as you are definitely actively building that bridge between the lab and the classroom. It's great. So in just a moment, everyone, we'll head to the breakout rooms, but first, a few notes. We understand that FEMA is conducting a test of the emergency alert system around 11:20 today. So if your cell phone goes off, just know that that is a test of the emergency alert system. It will be a little alarming potentially. And since you're automatically here, you are automatically registered for our Spring Professional Learning Network, which is on March 6th. And Dr. Tim Odegaard is going to give the keynote on that date. And then finally, we want everyone to save the date for our California Dyslexia Initiative Conference, which is going to be held virtually June 11th and 12th. And Jason will be there also, so that's exciting. So now I'm going to invite you to head to your breakout sessions. If you built a schedule on the Event Mobi homepage, you will see a schedule. If you did not, you just select sessions on the left side of your homepage and then choose the session you want to go to. Enter the Zooms by selecting Join Now, and it will take you straight to your breakout session, which starts at 10:10. We have 2 rounds of breakout sessions with 10-minute breaks in between. And once the final breakout is over, please come back to the closing session. We do encourage you to complete the feedback session, the feedback surveys, because it helps us as we plan upcoming events. And with that, I would like to say goodbye and please enjoy your morning.